In this presentation, we'll learn about some extensions to Mendelian genetics. These are situations in which the genetics does not seem to follow the patterns that Mendel described. In the presentation on Mendelian genetics, we looked at traits that showed what we call complete dominance. When we look at traits with complete dominance, we see that the heterozygotes look exactly like one of the homozygotes. Phenotypes are exactly the same, and we say that one allele is dominant to the other because the phenotype caused by the dominant allele hides the phenotype caused by the recessive allele. However, there are a number of situations in which the heterozygote does not look just like one of the homozygotes. When we looked at how the gene works, we found that the allele for purple flowers codes for an enzyme that is able to synthesize purple pigment. The recessive allele has a genetic difference that causes any protein that's formed to be unable to synthesize that purple pigment. As a result, heterozygotes still have enough of the enzyme to produce purple pigment, and that results in purple flowers. In other words, the presence of one allele produces enough enzyme to produce the purple pigment, and so the purple allele is dominant, and heterozygotes look exactly like dominant homozygotes. In some situations, the phenotype of the heterozygotes is not the same as either homozygous phenotype. With incomplete dominance, the phenotype of the heterozygote is somewhere in between the phenotypes of the homozygotes. This is the case with these snapdragons. If you cross red and white snapdragons, you'll get pink offspring. If you self these pink F1s, however, you'll get a mixture of 1/4 red snapdragons, half pink snapdragons, and 1/4 white snapdragons. The snapdragons have an allele that produces an enzyme that allows the flower to produce red pigment. The other allele fails to make red pigment, and so the flowers are white. Heterozygotes have a copy of each allele, and therefore they can produce some, but not as much, red pigment. As a result, the color is intermediate between the red and the white, and is pink. In all of these cases, the inheritance of the alleles is exactly as Mendel would have predicted. The difference comes in the relationship between the genotype and the phenotype. In this case, it takes 2 red alleles to produce a red flower, 2 white alleles to produce a non-pigmented white flower, and the presence of only 1 red allele will cause the flower to be pink. Notice that we avoid using the symbols that we previously used. Uppercase and lowercase symbols are used primarily to indicate dominance, and here we don't have dominance, so we choose different symbols. The MN blood types show an inheritance pattern called codominance. The M and N blood types refer to molecules on the surface of red blood cells that indicate to your immune system that the cell is, in fact, part of you and is not foreign. Your immune system will attack foreign cells, so it's important that your cells are marked as being part of you. Individuals with the M allele will place an M antigen on the surface of their blood cells. Individuals with the N allele will place an N antigen on their surface. And heterozygotes will place both M and N antigens on the surface of their red blood cells. The important thing to note here is that the heterozygote MN does not have the same phenotype as either homozygote, and the MN phenotype is also not intermediate between the 2 M and N phenotypes. Using a Punnett square, you can see that 2 MN parents could produce type N, type MN, and type M offspring. When we think about whether something is dominant or not, we need to think about the level of organization we're looking at. Tay-Sachs disease is an inherited disease in which the brain can't metabolize certain lipids because it lacks the enzyme to do so. As a result, the lipids accumulate in the brain and leads to the loss of motor and cognitive ability. At the organismal level, Tay-Sachs disease is recessive. Only individuals that have inherited both mutant alleles have the disease. Heterozygotes are completely normal phenotypically, and so we would consider this to be a recessive allele, and the normal allele to be dominant. However, if we look at the biochemical level, we see that only half as much enzyme is being produced by heterozygotes. Therefore, biochemically, this looks like incomplete dominance, with the heterozygote being intermediate between both homozygotes. Finally, if we look at the molecular level, then we see that both the functional enzyme and a similar but non-functional enzyme are produced. Thus, at this level, this looks like a case of codominance because the products of both alleles are present. In some cases, in fact in many cases, there are more than two alleles that are possible. In the ABO blood types, there are three common alleles in our population. The IA allele causes the cell to put a molecule on the surface of the red blood cells in much the same way as the MN blood types we looked at previously. Cells with only A-type molecules on their surface result in an individual having blood type A. The IB allele causes the cell to place B-type carbohydrates on its surface, resulting in type B blood in homozygotes. And the small I allele results in no carbohydrates being put on the cell surface. Let's start by looking at the relationship between the small I allele and the other alleles. This relationship is one of dominance. Whenever the small I allele is combined with an IA or IB alleles, the phenotype is of the dominant IA or IB alleles. Heterozygotes look like the dominant homozygotes, and we therefore say that IA and IB alleles are both dominant to the small I allele. Furthermore, because the small I allele doesn't cause any carbohydrates to be placed on the cell surface, small I homozygotes have blood type O. The relationship between the IA and IB alleles is one of codominance, just like with the MN blood types. IA-IB heterozygotes have a phenotype that includes both type of carbohydrates on their cell surface, and this results in the blood type AB. As a brief aside, you can see why some blood types are considered universal donors and others universal recipients. An individual's immune system is expecting to see either the A, B, or no antigens on the surface of red blood cells. An individual that has type AB blood will not mount an immune response if A, B, or even no antigens are on the surface of the cells. Therefore, we consider type AB blood to be the universal recipient. Individuals with type A or B blood accept type O blood because it doesn't have any antigens that the person's immune system is not expecting. Type O blood is considered to be the universal donor because any blood type can accept type O blood since there are no antigens to stimulate the recipient's immune system. Before we leave the idea of dominance, I wanted to briefly talk about what dominance is not. First of all, dominance has nothing to do with how common an allele is in a population. For example, the allele that causes achondroplasia and gives some people short stature is dominant. Therefore, heterozygotes will show the achondroplasic phenotype. However, the allele is very rare in the population, and so few people have the disorder. Secondly, dominance has nothing to do with what is considered to be the normal phenotype. Again, achondroplasic people are considered to have an abnormal phenotype, yet the allele is dominant. So just remember this: dominance only refers to the phenotype of heterozygotes. If the heterozygotes look just like a homozygote, then there is complete dominance. If the heterozygote is intermediate between the homozygotes, it's incomplete dominance, like with the pink snapdragons. If the heterozygote has features of both homozygotes, like with the MN blood types, it is codominance. Often, a gene has many effects on the body rather than just one. For example, Marfan syndrome is caused by a single allele, but results in phenotypic findings that affect many different parts of the body because many of those parts are affected by connective tissue that's the direct result of the allele that causes Marfan syndrome. Some people think that Abraham Lincoln had Marfan syndrome, though we don't know for sure. This effect of one gene on multiple aspects of our body, of our biology, is called pleiotropy. Sometimes a single trait can be affected by more than one gene. In epistasis, the effect of one gene alters the effects of the second gene. This is the case with coat color in Labrador Retriever dogs. The B locus determines the color, and so dogs may be either black, with a dominant B allele, or brown, with two recessive little b alleles. However, there is another gene at the E locus that determines whether the pigment is deposited onto the shaft of the hair or not. The dominant allele allows the pigments to be deposited, but the recessive allele results in no pigment being deposited. A little e homozygote will have no pigments deposited and will appear as a yellow lab, regardless of the alleles at the B locus. We can look at a dihybrid cross between two black labs that have these genotypes. The resulting offspring will be 9 black labs who all have both the dominant black coloration and the dominant E allele, allowing the pigments to be deposited. 3 of the offspring will be chocolate labs because they're homozygous recessive for the color allele, but they still have the dominant E allele, allowing them to deposit that brown color. 3 of them will be yellow because even though they have the dominant black allele, the homozygous recessive E allele prevents any pigments from being deposited. The last phenotypic group, the double homozygous recessive, will also be yellow because the homozygous allele, E allele, prevents the brown color from being deposited. Therefore, we have a modified 9:3 to 3:1 ratio that is modified to 9:3 to 4 by combining those last two groups. Sometimes there may be many genes that affect a particular trait. Often, these are traits which vary continuously in our population. In other words, there aren't discrete phenotypes. One example is height. There are people of all heights, not just discrete categories. Skin color in humans is another good example. To simplify, consider that each of the genes may contribute to more melanin being deposited in the skin cells. For each gene locus, there are some alleles that contribute more and some alleles that contribute less to this melanin deposition. For this simple example here, imagine 2 parents that are heterozygotes for each of these 3 genes. The uppercase allele contributes to melanin deposition, and the lowercase recessive allele does not. There are many genotypes that the offspring may be, and some of them involve many of the dominant alleles, resulting in dark skin, and some of them involve many of the recessive alleles, resulting in light skin. Although many skin tones are possible, the light and very dark skin tones result from relatively uncommon combinations of alleles. The intermediate combinations, however, are much more common because there are many ways of creating them genetically. This normal distribution, or bell curve, is really common for polygenic traits like this. Finally, there's also often a strong effect of the environment on phenotypes. These hydrangeas have exactly the same genotype. They're clones of one another, but very different phenotypes depending on the acidity of the soil they're grown in. We usually think of human height as being an example of a polygenic trait, and indeed it is. However, height is also dependent on our nutrition and the interaction of our diet and our genotype. Individuals with a healthy diet are able to grow taller than those with an unhealthy or limited diet, even given the same genetic makeup. When a trait is influenced by the environment like this, we say that it's multifactorial.